Chapter 11: A Fountain of Life. In its myriad forms, life is both a marvel and a mystery. It represents a journey through physics and chemistry into biology, a field brimming with complexities and perplexities. From the simplest cellular organisms to the most intricate multicellular beings, the phenomenon of life unfolds in ways that continue to intrigue scientists, philosophers, and thinkers alike. Yet defining life remains an enduring challenge. Is life solely defined by the presence of cells, or can it also encompass entities that blur the boundaries, such as viruses and plasmids? This chapter delves into these profound questions, exploring the properties that distinguish living entities from inanimate matter, and examining how life manifests at cellular and acellular levels. The quest to define life is central to our discussion, a pursuit that reveals its intricacy and diversity. We explore the idea that life is the logic of existence via propagation, emphasizing that the continuity of life hinges on its ability to sustain itself and transmit its essence across time. But is life's purpose bound to the here and now, rooted in spatial objectives, or does it transcend into temporal aspirations for longevity and evolution? Since my school days, the question of what defines life has stayed with me. I vividly remember Dr. Ashok Kumar, my biology teacher, engaging us with concepts like reproduction, response to stimuli, metabolism, and other attributes that attempt to define life. Those thought-provoking discussions sparked my enduring quest to unravel the essence of life. In this chapter, we shall reflect on the logic of life, characteristics, and objectives. Through this journey, we aim to illuminate the essence of life as a fountain of existence and continuity, cellular beings and acellular propagating entities. Life must have originated from matter, whether on Earth or some other planet. Matter must have transformed into something with lifelike properties, which eventually gave rise to protocells, the earliest forms of cellular life, bacteria, and then multicellular organisms, eventually leading to humans. So what is the specific point at which we consider entities to be living? Most scientists, philosophers, and people would define entities consisting of cells as life. There are also other propagating entities that are not cells. We will refer to these two categories as cellular entities and acellular entities. Cellular propagating entities include bacteria, archaea, protists, fungi, plants, and animals. Acellular propagating entities include viruses, plasmids, and viroids. Acellular propagating entities do not possess life properties such as metabolism, respiration, and response to stimuli. The only living property they have is reproduction, and even then, they can only reproduce as parasites within other cellular propagating entities. Viruses consist of nucleic acid, genome, and proteins. Viruses are referred to as semi-living or conditionally living. They are considered living inside the cell, but non-living outside the cell. Plasmids have the same properties as viruses, except that they lack proteins in their physical structure. Then there are viroids, which are the simplest single-molecule propagating entities. To determine the essential properties and hopefully aid in the definition of life, let us compare some specimens consisting of acellular entities and cellular entities. Acellular entities are viruses, plasmids, and viroids. Cellular entities include animals, plants, bacteria, fungi, and protists. All the cellular forms mentioned exhibit metabolism, growth, and reproduction, and contain genetic information. Acellular forms do not exhibit metabolism and growth. However, they propagate through the replication of their genetic material. Therefore, it seems that the essential properties of living entities are having genetic material and the ability to reproduce, propagate. To further complicate matters, let us consider the case of seeds, spores, bacterial, fungal, protozoan, eggs, a mule, a sterile offspring obtained through interbreeding a horse and a donkey, and human cells grown in culture plates, artificially in a lab. In these special cases, seeds and spores are in a dormant state, which is similar to how viruses are when outside cells. Seeds and spores can give rise to an organism, but whether we consider them living or dead mostly depends on their potential to give rise to an organism. For the sake of progress, let us consider them living. A mule, or any sterile individual, exhibits metabolism, growth, homeostasis, motility, response to stimuli, genetic information, complexity, and death. The only characteristic that a mule does not exhibit is reproduction. A mule cannot reproduce, but the cells within it can give rise to more cells. It has cells that can form new entities within the same organism. Cultured human cells in the lab exhibit all the characteristics, including giving rise to new cells, although limited, and are therefore considered living entities. However, they are not organisms. The only common factor among every entity considered in this section is the presence of genetic material with the potential for replication. A mule or cells in a culture flask can still replicate their genetic material. The ability to replicate genetic material is the only shared feature among all propagating entities. The inherent ability to replicate genetic material defines a bioentity or a living entity. Properties of living things. Metabolism. Metabolism refers to the interconversion of matter and energy within a system, utilizing energy input to generate usable molecules and energy. It is divided into two components: catabolism, the breakdown of molecules, and anabolism, the building of new molecules. In living organisms, metabolism involves the use of chemical or light energy for essential life functions such as growth, motility, and reproduction. For example, a cheetah consumes prey, digests the flesh to release chemical energy, and then converts that energy into mechanical energy for chasing new prey. Similarly, a car uses petrol to generate energy, releasing heat and gases in the process, which results in movement. However, there is a fundamental difference in how the input matter exits the system in these two cases. In a car, the combustion of petrol produces mechanical energy, and the byproducts of this process, such as heat and gases, are released immediately. In contrast, in living organisms, the matter is assimilated, integrated, and used for constructing or maintaining the organism's structure. The products of metabolism in living organisms become part of the organism itself. They are not discarded immediately, but are employed in building the body or replacing other molecules. Unlike a car's metabolism, which primarily involves the breakdown of materials, the metabolism of living organisms involves both the breakdown of molecules and their synthesis into new ones. Living organisms are capable of anabolic processes in which they synthesize new molecules from simpler components. This anabolic capacity allows them to harvest energy and assimilate matter, resulting in growth and development. Few non-living entities, such as solar panels, exhibit true anabolism. In living organisms, metabolic processes are distributed throughout the body, as metabolism occurs in almost all cells, tissues, and organs. These processes occur at both the molecular and cellular levels, and the energy and matter flow within the organism to sustain life. Non-living objects like cars do not exhibit a similar thermodynamic state. While the car may have fuel that represents a higher thermodynamic state, this state is externally supplied and does not arise from within the system. Moreover, very few parts of the car demonstrate molecular-level dynamism. The regulation of a car's metabolism is governed by human input or programming, in contrast to the natural regulation of metabolism in living organisms. Organisms vary in terms of metabolic activity, with tissues showing different levels of metabolic dynamism. The extent of metabolism in an organism can be assessed by the proportion of protoplasm, the living material of the cell, relative to the whole body. Protoplasm consists of the cytoplasm, nucleus, and other organelles where metabolic reactions primarily occur. For instance, leaves rich in protoplasm undergo more metabolic activity compared to stems, which contain less protoplasm. In humans, tissues with high metabolic activity, like muscle or liver tissue, contain abundant protoplasm, while bones and nails have far less. The protoplasmic factor, defined as the weight of protoplasm relative to the total body weight, can be used to assess an organism's metabolic capacity. Plants tend to have a lower protoplasmic factor because much of their body mass consists of structural materials, such as cellulose and lignin, which are less metabolically active. Consequently, plants often survive longer than animals. For example, when a tree dies, the leaves are the first to be lost, whereas the more structurally supportive parts, like the veins of a leaf or the trunk, persist longer. The longevity of organisms is influenced by their metabolic activity. Those with lower metabolic rates and protoplasmic factors tend to last longer. While there are exceptions, the correlation between metabolic activity and survival is clear. Higher metabolic activity often correlates with shorter lifespans, while slower metabolic processes contribute to greater longevity. Oxidative metabolism is likely to cause more genetic damage while all else is constant. Growth. Growth is defined as the change in the occupancy of space due to an increase in matter and energy. It represents the net increase or decrease in mass. When the input of matter exceeds the output, the entity's mass increases, resulting in growth. Conversely, if the input of matter is lower than the output, the entity shrinks. All living organisms undergo growth at some point, but non-living things can also grow under specific conditions. For example, a salt crystal can grow when placed in a supersaturated solution, or an ice crystal can form in freezing conditions. Similarly, the rusting of iron, the aggregation of molecules like proteins and lipids, or the formation of double-stranded DNA from complementary single strands are all forms of non-living growth. When a supersaturated sodium acetate solution is poured onto sodium acetate crystals, the crystals act as nucleation centers, and more sodium acetate is deposited, releasing heat in an exothermic reaction. These examples demonstrate that growth in non-living things often occurs due to environmental conditions rather than the intrinsic properties of the entities themselves. The growth of living organisms, however, differs fundamentally from that of non-living things. To understand the distinction, one must examine the mode of growth, the energy required, and the assimilation of matter. Growth in non-living things, as illustrated by crystal formation or droplet condensation, is often exothermic and dependent on the environment. In these cases, the environment facilitates the addition of material to the growing entity without the entity inherently attracting or assimilating the material. For instance, water droplets condense on a cold surface where the molecules are added due to cohesive forces, and crystals grow when environmental conditions exceed the material's carrying capacity. This type of growth is superficial, with the material simply adding to the surface. In contrast, growth in living organisms requires energy. Organisms derive matter and energy from their environment, using this input to grow in size. This process is not spontaneous. It requires considerable energy expenditure. Organisms grow despite environmental challenges, such as the development of bones, shells, or exoskeletons. Unlike the crystal growth in non-living things, which is driven by external factors, the growth of living organisms is controlled internally. It follows a definite inherent pattern dictated by genetic information. Hippocrates believed that magnets possessed human-like powers, and I, like many, find magnetism fascinating. A magnet picking up iron filings is often considered a form of growth. However, this is not true growth in the biological sense. While a magnet can attract and accumulate iron filings, it forms a different shape each time it does so. Additionally, the magnet remains the same if the iron filings are removed. The magnet and the iron filings remain independent entities. On the other hand, in living organisms, the matter that is assimilated becomes a part of the organism's structure. The assimilated material is not merely added to the organism's surface. It is integrated into its molecular structure and used to build cells, tissues, and organs according to genetic information. Unlike non-living growth, where the added material can be removed without altering the entity, removing assimilated material from a living organism would disturb its integrity. Growth in living organisms is characterized by the assimilation of simpler molecules that are then used to create complex structures. For example, when I consume a banana, the matter and energy from the banana are digested, absorbed, and incorporated into my body. The material is not just added to my body's surface, it is used in cellular processes and contributes to the growth of new cells. In this way, the matter becomes an integral part of my organism, taking on the properties and characteristics of the entity. Growth can also be understood in terms of how the occupancy of space changes. In unicellular organisms, growth occurs through an increase in cell size or cell number, with asexual reproduction often resulting in the division of the cell into two. In multicellular organisms, cells are bound together and differentiate individually or through coordinated activity. Over time, as the organism grows, its size increases, reaching its carrying capacity based on the availability of matter, energy, and the niche in which it resides. Reproduction. Reproduction, in its simplest sense, refers to copying something. In biology, An organism can generate another organism with similar features and identity rooted in the genetic information that governs its structure and function. Reproduction involves creating more copies of entities such as organisms, cells, viruses, plasmids, transposons, and viroids. It recapitulates the parent's physical form and genetic makeup in each successive generation. The process results in one or more new entities with similar properties, with the potential for continued existence and further reproduction. Reproduction is driven by acquiring matter and energy, not loss. Growth, which precedes reproduction, allows an entity to reach the potential required for reproduction. Reproduction should not be confused with simple division, where there is a loss of matter and no potential for regrowth. For example, if a brick breaks into two pieces, it forms two separate entities, not a reproduction. The newly formed pieces lack the potential to regenerate the lost matter. Similarly, a magnet that is broken into two pieces still retains its north and south poles, but neither of the pieces can regrow to the size or strength of the original magnet. A computer virus replicates within a system and spreads across the internet, but it remains immaterial.